Hi, welcome back, everyone. Uh, we are going to have the first talk of the day for day 3 of the Human Enhancement Conference. And we are going to start with a keynote speech from Michael Levin, a really big figure in the space of longevity and bioelectricity, who is known for his pioneering work on xenobots, living robots made from frog skin cells, and also for his contributions to the field of bioelectricity. So I'm super looking forward to this talk, and I hope you are too. So, Michael, feel free to start.
Thank you very much. And thank you for having me here. My name is Mike Levin. I'm the director of the Allen Discovery Center at Tufts, and I'm a developmental and regenerative biologist. And what I want to talk to you about today is the plasticity and the potential for learning to engineer the agential material of life. And if you're interested in these topics, you can find all of the information here. I want to start by thinking about the end game of our field, and I call this the anatomical compiler. Someday, you should be able to sit in front of a computer and draw the plant, animal, organ, or biobot that you want at the level of anatomy, and the system would compile your description down to a set of stimuli that would have to be given to cells to get them to build exactly what you want them to build. In this case, this nice three-headed flatworm. Now, on a practical level, why do we need this thing? Well, if we had the ability to control what cells build, then all of these problems here would go away. Birth defects, traumatic injury, cancer, aging, degenerative disease. None of these things would be an issue anymore if we had the ability to communicate goals to groups of cells. Now, this is profoundly difficult. We are still far away from this, even though molecular biology and genetics have been going very strong for decades. Why is that? Why don't we have something like this? And the fact is we are very good at molecular kinds of information, figuring out what genes turn on other genes and things like that. But we're really very far away from a large-scale control of form and function, such as restoring damaged organs and longevity. And I think that's because biomedicine has been operating largely at the level of hardware. And we can see what this looked like for information technology. Back in the 40s and 50s, this is what programming looked like. You had to interact directly with the hardware. There she is rewiring the machine. And all of the most exciting advances today in biomedicine are things like genomic editing, protein engineering, pathway rewiring, and so on. These are all down at the level of the molecular hardware of life. And I think what's going to happen in biology is exactly what happened in computer science when it was understood how to program devices without having to rearrange the hardware. The software of life, and in particular the intelligence of the living material, is going to open some really fundamental new approaches in regenerative medicine and beyond. We all used to be single cells, so we want to take a look at the kind of material that we are made of, and this is what we are made of. This is a single cell. Now, this happens to be a free living organism called a lacrimaria, but you can see what's happening here. It has no brain, no nervous system, and yet it is incredibly competent at its local single cell agendas. And we all started life as a single cell like this, the unfertilized egg, and eventually we became one of these remarkable things, or even something like this. And so we are composed of material that is already very capable of certain things. In fact, even inside of cells, you have molecular networks like this. And these molecular networks, as we have shown, are capable of 6 different kinds of learning, including Pavlovian conditioning. And in our group, we make devices such as these to try to train cells, in fact, to try to train the material inside of cells for purposes like drug conditioning and all of that. And this is all described here. So what the human body, or in fact any body, consists of is a multi-scale competency architecture. At every level, there is a problem-solving capabilities, from the molecular levels all the way up through tissues and organs and organisms and even swarms. These systems have the ability to navigate their own problem space and to solve problems. So this is very different from the kind of engineering that we've been doing for thousands of years, where the material is passive and you control it by determining every aspect of function. This is completely different. The biology requires a different approach. And I'll just show you one simple example of this kind of problem solving. These tadpoles, so this is a tadpole of the frog, and they have to become a frog. And in order to do that, they have to rearrange their face. So the eyes have to move, the nostrils have to move, the mouth, everything has to move. And it was thought for years that this was some kind of hardwired process, that every organ just moves in the right direction and in the right amount, and then you get your frog. So we decided to test this, and we found that if you scramble these initial organs, so here's one of our Picasso tadpoles, the eyes on the top of the head, the mouth is off to the side, everything's scrambled. You don't, what you get is a perfectly normal frog. And that's because all of these organs, even though they're starting in the wrong position, they will move through novel paths to get to where they're going, and then they stop. So this ability to meet your anatomical goal despite novelty, despite different starting positions, is just one example. There are many examples of this kind of competency of the material. And so the genetics does not actually specify some kind of hardwired outcome. What it gives you is a flexible problem-solving system. And now you might ask the question, so how does this work? How do these tissues know what a correct frog face is supposed to look like? How do they know where to go? Okay. And we've been studying this, and it turns out that the way it works is pretty much the same, using the same mechanisms by which organisms know what to do to meet their goals. In other words, the same bioelectric mechanisms that are used in the brain. So in the brain, you have the hardware, which is a bunch of cells connected in the network by these little electrical synapses, and you have these ion channels that allow charged molecules to go in and out, and that sets a voltage across the membrane. And as these voltages propagate through the neural network, you get this. This is what it looks like in a living zebrafish brain, and it is the commitment of neuroscience that all of these electrophysiological events that you're seeing here could be decoded to tell you what the fish is thinking, what memories it had, what the goals are, and so on. But it turns out that this amazing system of using electrical networks to store and process meaningful information within groups of cells is actually extremely ancient. Evolution discovered this way back at the time of bacterial biofilms. And actually every cell in your body has ion channels. Most cells have these electrical connections to their neighbors, and the exact same kind of computations that are going on in your brain are going on in the rest of the body. Except instead of thinking about how to move you through three-dimensional space towards your goals, what are these networks thinking about? They're thinking about shape. They're thinking about three-dimensional structure. And we've developed some technologies for being able to read the electrical messages that the cells are sending to each other, just like neuroscientists image in the brain. And so here you can see an early frog embryo, and here all the colors are showing you all the electrical conversations that these cells are having with each other. And this is not a model. This is fluorescent voltage reporting dyes used in real tissue. So these pattern memories, these are literally memories that tell the cells what they're going to build. I just want to show you a couple of examples because we can now visualize the cognitive content of body cells. So this is a time lapse of an early frog embryo putting its face together, and you can see here. This is one frame out of that movie. Here's where the eye is going to be. Here's where the mouth is going to be. Here are where the placode structures. So we can literally now read out the subtle bioelectric scaffold that tells the different organs where they're going to be and turns on and off the different gene expression and so on. Not only is the bioelectricity a kind of cognitive glue that holds together individual cells towards the global goal of making a proper face, just like it acts as cognitive glue in your brain to make sure that you and I are more than just a pile of neurons. It actually works that way between organisms too. So you can see here, each one of these things is a separate embryo. And when we poke this one in the middle, it has this calcium wave that actually all the neighboring embryos find out about it. Okay, and so you can see here, this is these cells. Each one of these things is a separate cell. And again, you can see all the electrical activity. There are some neurons here firing, but also all this other stuff that is not neurons is processing information as well. So now that we've learned to read this electrical information, we can also write it. And this is important. We do not use any kinds of magnets. There are no electrodes. There are no frequencies, no waves, no electromagnetic fields. What we do is we manipulate the ion channels and the gap junctions. In other words, we control the interface at the edge of these cells that the cells are normally using to control each other. That's why all this works, because we are hijacking a native system of computation and memory within the tissue cells. And I'm going to show you very quickly a few examples of what do you gain when you have access to this interface. So we created these molecular tools to be able to read and write pattern memories into tissue. And so why would you want to do that? Well, one example it has to do with cancer. And that is that when we inject human oncogenes into these tadpoles, here's a tumor, but even before that tumor becomes apparent, you can already see these cells, due to the action of that oncogene, these cells are starting to disconnect. They have an abnormal voltage. They disconnect from the electrical network. They no longer remember what they were building, and they go off basically like amoebas, treating the rest of the body as just external environment. And for this reason, we can think of cancer as a kind of dissociative identity disorder. It is these cells are not more selfish than normal cells. They just have smaller selves. As far as they're concerned, each individual cell is out for itself, and the rest of the organ is just outside environment. But normally, they're part of this big network. And the network as a whole has very grandiose goals of building nice organs, nice skin, nice muscle, and so on. So once you know this, what you can try to do is not kill the cells, but force them into proper electrical connection with each other. So when we inject this oncogene, we can also provide an ion channel RNA that specifically forces these cells into the correct bioelectrical state. So it prevents this. And here, this is the same animal. Here's what you see. The oncoprotein is strongly expressed in red here. In fact, it's all over the place, but there's no tumor. And the reason there's no tumor is because it's not the genetic defect that drives the outcome. It's the decision-making of the cells. And in this case, the cells are part of a network that does not want to make a tumor. It wants to make healthy skin and muscle and so on. So the first application is a normalization of cancer, and we are now moving on to human tissues. Having done this in frog, we are currently working on this in human tissues. The next example is a repair of birth defects. So here's the brain of an early tadpole here, and you can see forebrain, midbrain, and hindbrain. And there are a variety of teratogens, including chemical and mutations. In this case, mutation in the gene called Notch. This is a very important gene. If you mutate it, the forebrain is gone. The midbrain and hindbrain are just a bubble. These animals have no behavior. They're profoundly affected. And what we know is that actually the brain is self-constructs by virtue of this bioelectrical pattern. Here it is in more detail. This is the pattern that tells the brain exactly what shape and size to be. We made a computational model of this, and we asked the model, if the pattern is incorrect, such as in this case, what channels could you open and close to make them correct? And the model told us that there's a very specific ion channel called HCN2, which can be used to repair such defects. We found a drug, in fact, a couple of drugs that open HCN2. And what you see here is that they actually restore the structure of the brain despite this incredibly powerful mutation. And in fact, these animals have normal IQs. Their learning rates are indistinguishable from controls, even though they have the same mutation. So at least some hardware defects, and this is a Notch mutation, so this is a hardware defect. Some hardware defects are fixable in software. So this is an example of the use of, by the way, already human approved. So these are anti-epileptics that are already in human use. Our computational model is able to find human approved electroceuticals which repair the bioelectrical pattern, and the cells follow right after that. And the final example I want to show you has to do with regeneration. So in adult frogs, if they lose a leg, they normally do not regenerate their legs. So 45 days later here, there's basically nothing. We developed a cocktail that says to these cells, go down the leg building route, not the scarring route. And what happens is, after just a very brief application of this cocktail, immediately the pro-regenerative genes such as MSX1 start to come on, and within 45 days you've already got some toes and you've got a toenail. Eventually you get a very nice leg, which is both touch sensitive and motile. And what's happening here is not that we're controlling stem cells. There are no scaffolds. In our latest experiments, the treatment is 24 hours, after which the leg grows for a year and a half. You get a year and a half of leg growth after communicating very early on to the cells that they need to regenerate, and that's it. We don't touch the system after that at all. The goal is not to micromanage gene expression or cell differentiation or anything like that. The goal is to communicate using this electrical interface very early on what the cells should be building. And so at this point, I have to do a disclosure because David Kaplan and I have this spinoff called Morphoceuticals, which is now moving this into mammals and hopefully eventually into human patients. And then the next example I want to show you is being able to produce whole ectopic organs. So here's a tadpole, and what you'll see is here's the normal eye, but you'll see there's actually an extra eye on its gut. And so how did this thing get an extra eye, which, by the way, has all the normal lens, retina, optic nerve, all the normal components. It's because what we did was we injected an ion channel RNA that creates a little spot that's just like that spot you saw in the electric face at the beginning of the talk. It's a voltage pattern that tells the cells build an eye here. And so when they see that pattern, they build this eye. We don't have to micromanage this process. We don't tell them how to build an eye. In fact, we have no idea how to build an eye. But what we have found is this bioelectrical subroutine call that says build an eye here, and then the cells being competent material, the cells do everything else, all the gene expression, all the cell differentiation, all of that. And so what I've shown you is the ability to repair and induce and regenerate normal structures. And now in the last couple of minutes, I want to go beyond the standard structures of the body and think about the plasticity of the future. So beyond just the repair of what normally is there. Embryonic development is extremely reliable. And so everybody sees these acorns and they see the oak leaves that they make. And we sort of get this idea that this is what the oak genome knows how to do. It knows how to make this particular structure, and that's it. And we would still be thinking that, except that along comes this non-human bioengineer, so this little wasp. And what the wasp has learned to do is to hack the morphogenesis of the leaves. In other words, it leaves some signals that actually cause these flat green cells to build something like this or something like this. These are called galls, and they're incredible, complex, beautiful structures. We would have absolutely no idea that the cells that build this so reliably build this flat green thing are capable of even building something like this. And in fact, the sophistication of what can be built somewhat matches the sophistication of the engineer. So bacteria make these kind of featureless blobs, fungi do a little better, nematodes a little better, and then by the time you get to insects, you get these beautiful structures. And so we can see that life is eminently reprogrammable. It is the genetically specified hardware can do many interesting things. And the very last thing that I want to show you is an example of the synthetic biobot. So you heard about our xenobots, which are biobots made for environmental applications made out of frog skin. But I want to show you something regarding biomedicine. This little thing right here, it might look like a creature we got from the bottom of a pond somewhere. But actually, if you were to sequence the genome, you would find this is 100% normal Homo sapiens. These are human, adult human tracheal epithelial cells. There's nothing embryonic here. These come from adult patients who donate tracheal samples. And we found a way to have them self-assemble into this little creature. And this looks like no stage of human embryonic development. This is completely novel plasticity. And one of the things it can do, for example, if you have neurons here and you put a big scratch through it, these are human neurons, you make a big wound, these biobots will land in the wound, they will assemble into a superbot cluster. And what you see it starts to do after about 4 days, you can see if you lift them up, you can see what it does is it heals the wounds. So who would have thought that your tracheal cells that sit there quietly in your airway are actually capable of forming a self-contained little creature that runs around and by the way, is capable of this healing. So this is just we're just scratching the tip of the iceberg here. They probably have many more capabilities, and we envision them as personalized autonomous therapeutics. In other words, they come from your cells. They're not genetically modified. There are no weird nanomaterials or scaffolds or drugs or anything like that. They can be put back into your own body with no immune suppression needed. They live for a few weeks and they can exert reparative effects. So we think this is the beginning of a new personalized technology that doesn't use genetic modification or foreign materials to exert repair in the body. So I'll just end here by pointing out that I think future medicine is going to look a lot more like a kind of somatic psychiatry than it's going to look like chemistry. Right now, everything is focused on the hardware. Okay? So all of these kinds of approaches, and they do many useful things, but there's actually lots and lots of opportunity here in the space of top-down controls, including training cells and tissues, including agential implants such as Anthrobots, and of course the electroceuticals that I showed you today. And we're working on applications to use AI to communicate to these various systems of the body, because the future medicine is not about micromanaging molecular states. It's about collaborating with and communicating your goals to the material of the body, which is actually quite good at carrying out various goals. So learning to rewrite it is critical. And because of this, we have an interesting future because basically, because of the plasticity of life, any combination of evolved material, engineered material, and software is some kind of agent. We already have hybrids and cyborgs and people with all kinds of implants and augmentations. Potentially, the space of possible embodied minds is enormous. all of the natural world that Darwin saw when he said endless forms most beautiful is like a tiny little region of this space, these creatures that we're going to be living with in the coming decades. And I think it's really important to understand the emergent intelligence of novel material, whether biological or engineered, and think about how we're going to have a kind of synthbiosis with these other beings. So I'm going to stop here and just thank all the postdocs and students that did some of the work that I showed you today. We have lots of amazing collaborators. These are the funders that we've had over the last 25 years, and here are 4 companies that we are involved with. So thank you very much, and I will take questions.
So much, Michael. So by the way, we actually have 1 hour reserved for this session. So feel free to expand at any point. Maybe if you get like a question, like, you know, any specific subsection, like feel free to be a little bit more lengthy with your answers, because we have until, 2 p.m. Roatan time. So I'll be taking questions. Any question? Okay, we have multiple ones.
Hey, Mr. Levin. Fascinating talk, absolutely beautiful.
Thank you.
I might have missed a very, very small part of it. So just to very quickly recapitulate, is it correct that you're essentially implying that the general structure, bodily structure, is organized through electrical activity and essentially almost this Venn diagrammatic intersection between cells?
Yeah, you can think about it very much like neuroscience. So in neuroscience, I'll just give you an example. There's a particular kind of magic that goes on every day, and you might think it's some sort of really exceptional experience of mind-body medicine. But look, when you wake up in the morning, you have these large-scale goals. You have social goals, career goals, all this very abstract stuff. But in order for you to get out of bed and execute on any of those goals, ions have to be pushed across your muscle membranes. In some way, calcium and potassium and other ions have to move. The biochemistry literally has to be responsive to these very high-level information processing tasks. And so what enables that to happen in the body, I mean, it's literally mind over matter. It's decisions that you make in a very abstract space have to eventually filter down to make the ions dance in your muscle cells. So that whole process is implemented by neural bioelectricity. In other words, when you have specific patterns of electrical activity in your brain, those eventually change gene expression, they change cell behavior, and they make things happen in your body with voluntary motion, gland activity, everything else. The same scheme works everywhere else. In order for your cells to know and to remember what it is that they are building, or to be able to rebuild it if it gets damaged, they need memories. And those memories are electrical patterns that are outside of the brain. They're present in all tissues. And the system of that we know by the brain and the nervous system is basically just an evolved, sped up version of what evolution was doing long before that, which is to use electricity to store large scale goals that coordinate cell behavior. So all of the things that you're familiar with, gene expression and the stem cell differentiation and the movement of signaling molecules, all of that stuff is in an important way downstream of decisions that were made in a proto-cognitive medium, which is the electrical activity of tissue.
Right. And so to follow this up, you mentioned that in your, I guess, perspective, the general disposition of the body is actually not determined by genes. It's, again, mainly determined by those very electrical signals. But then surely genes actually specify, for example, how many ionic channels, for example, potassium channels you might have in your neurons. Could that be actually the instruction set that then determines essentially the bodily disposition? Like, for example, the maybe over influx of potassium channels might, I don't know, give you too many fingers, for instance.
Yeah. So, okay, great question. So let's dig into that a little bit. And the right analogy to use for this, and I'm going to tell you why. But the right analogy to use for this is the analogy of the hardware and the software in the computer. In other words, if somebody says to you, "Yeah, you showed me an algorithm and everything, but look, the real thing that's happening here is the silicon, the copper, and Maxwell's equations." that is what's actually underlying everything that you're seeing. And so that's not exactly false when you take the kind of reductionist stance on these things. But the question to ask is, which of these paradigms enables you to make the most efficient change? So in other words, would you would never hire a coder that came in and said, look, I don't believe in these high-level algorithms. Everything is determined by Maxwell's equations and the way that electrons inevitably move through the medium. And the same situation is true here. You could, of course, none of this would work without the ion channels and such. So they are absolutely necessary. And what the genome does is make sure that every cell has the relevant hardware that enables it to do various things. But now let's ask the question: is the genetic level the optimum level of control here? Does it enable optimum prediction and control? And I'm going to give you a few examples that just show you why that's not the case. First of all, an example that I already showed you was in this. Let's see. In this case. So for example, if you were to sequence this animal, what you would see is a very strong oncogene, and you would make a prediction. The prediction would be that there should be a tumor there, and you would be incorrect because it isn't actually the genetic information that drives; it's the physiological information, and you can't derive that. The difference between this tissue and this tissue is not visible on any omics that you could do, because these channels open and close post-translationally. The genes are the same, the transcripts are all the same. It is not the case that you know where your depolarizations are based on where the channels are expressed. You have to do the physiology, just like in a computer. If I tell you if I give you an account of where all the silicon atoms are in RAM, you still don't know the content of what the information is there, because it's stored as patterns of energy through the system. It is not actually the hardware that determines it. Same thing here. If you were to sequence this animal, you would see the notch mutation, and you would say, well, we know exactly what happens when we see that notch mutation. We should have this defect. And again, you wouldn't see this because this was repaired not at the level of changing gene expression. It was repaired at the level of opening and closing ion channels, which control a bioelectrical state. And finally, there's an even more amazing example, which I didn't bring the slides here because I thought I only had 20 minutes, is the example of head determination in flatworms. So we got these amazing flatworms called planaria, which are, by the way, immortal. So it's an interesting model system for aging and longevity. They're immortal. And what you can do is you can cut them into pieces, and every piece regenerates a perfect worm. So they make a head at one end and the tail at the other end. So what we found is that we asked the question, how do they know how many heads to make? And what we found is that there's an electrical pattern that says one head, one tail. And we were able to rewrite that pattern to say two heads. And if you rewrite that pattern to say two heads, and then you cut the animal into pieces, each piece regenerates two heads. And then what you can do is what you can continue to recut those two-headed animals, and they will forever continue to regenerate as two-headed. We have permanently changed their target morphology, the pattern to which they regenerate upon damage. So now it's interesting. When we do this, we don't touch the genome whatsoever. Okay, this is purely done by rewriting the electrical pattern memory, the sort of volatile RAM, if you will. And so if you were to come across these animals, you would see these two-headed animals, you would see the one-heads, and you would say, cool, there must have been a speciation event. Let's sequence the genome and see where that speciation event took place. And you would sequence the genome, and you would find absolutely no differences. And so then now we can ask, is the number of heads, you would think that something as important as the number of heads would be encoded genetically. And it turns out that's not the case. What is the case is that what the genome encodes is a bunch of hardware that by default creates a pattern that indicates one head. Just like when you buy a calculator from the store, by default, you turn it on, first thing you see is zero. Reliably, you'll see a zero on the LCD. That's what the circuit does by default. But it's reprogrammable. That memory is reprogrammable, and the actual outcome is not determined by the hardware. That's simply the default behavior. You can put in a new pattern which says two heads, and it's just as happy to make two heads and there was no change to the actual hardware. The change was all at the electrophysiology level. So basically, I think that's the right way to think about this, is that what the genetics gives you is some very nice reprogrammable hardware. And by the way, some genetic mutations will absolutely mess up that hardware. So if you're missing some channels, or your channels are damaged, you will not be able to execute certain circuits that are necessary for certain behavior. I mean, this is true, but the capabilities of the system are by no means exhausted by the information that's in the genome.
Right, that's super clear. And maybe one last question before I actually pass it on. Would you recommend any literature on this subject? Maybe both of yours or of anyone else's that I could write down and actually dive into this a bit deeper, because this seems really, really fascinating.
Yeah, thank you. Drop me an email. I'm sure the organizers have my email. There are hundreds at this point of papers on this. I've given many long presentations on it. There's more information that you could than you could want on this. So just drop me an email, and I'll send you a link to. We have everything on our website. All the actually, wait a minute. Yeah, so if you go, so here's some websites, but drop me an email anyway, and I'll just send you some reprints. But if you go here, there's a publication page, and then some resources pages and things like that have all of the code, the data, the primary papers, all of that stuff is here. This is my personal blog, where I kind of talk about what all these things mean and kind of try to digest some of the papers in a more readable form for outsiders.
Incredible, fantastic. I'll send you an email. Thank you.
Perfect. Thank you so much.
Thank you so much, Michael. Yes, I will share the email, and we have at least 2 more questions. Let's go. Okay.
Hi, Michael, and thank you. Absolutely fascinating.
Thank you.
I'm Brent Streiner. I'm an investor, advisor, private equity guy, but not in this field. Could you just go back to the personalized autonomous therapeutic slide?
Absolutely.
Understanding the applications for this would be to...
Let's see. Is this the slide you wanted, or you want to see the actual Anthrobot?
No, it's the other one where, yeah, that one is specific to neural repair. But am I correct in understanding that the applications for this are absolutely vast? And if
I would, yes. Clearly, I'm not a disinterested party in this question, but yes, of course, I think the applications are incredibly vast. I mean, look. The reason I'm showing you this is this is the first experiment we did to see what they can do. This is not part of a large screen where we tried 800 things and this was the only thing they could do. This was the first thing we tried. Okay, so I don't really believe in luck to that level, and I think that if this is working out of the box, that probably means that there are hundreds of other things that they can do. We are now in the process of figuring out what some of those things are, but it's very clear that the material that comes from your own body has many healing competencies that we don't even begin to be able to predict. I mean, nobody could have I mean, this actually goes back to the previous question of the genome, right? I should have mentioned this. If you sequence the genome of this critter, the information you get has zero predictive value for the shape, the function, and the behavior of this thing. Because what you'll see is Homo sapiens, and you think you know what Homo sapiens genomes do. They make humans, and you would never guess any of this, and you would certainly never guess any of this. So that's another actually example of that. But yes, I think the implications are huge, because these things. I mean, just think about this. They compared to any other nanobot that you might make, when you build something out of passive materials, it's up to you to control every part of its function. This thing has over 1 billion years of experience with things like metabolism, stress, damage, cancer, infection, and so on. We don't have to build any of that in. And you have this incredible system that comes from your own body, so you don't need immune suppression. There are no worries about weird nanomaterials ending up in your body and so on. So yeah, I would agree with that. I think this is a foundational new approach to this, and then we have to now find out all the things that they can do.
Yeah, I mean, okay, so it's very early stage. It seems like a relatively recent breakthrough.
About a year. Yeah, this has been we've known this for about a year.
Please consider your personal security seriously.
I'm happy to take advice on that. I have no idea how one does that, or what the what are the steps. I have no clue.
It's trillions of dollars that do not want to see this type of technology released mass market. That's the only consideration.
I hear you. I understand what you're saying. I just don't know I'm an academic, and so I don't know what resources people use to ensure their personal security. I have no clue. So if you have thoughts, I'm happy to hear them on an email.
Okay, thank you so much. Okay, one more question over here.
Can you speak a little more to the translation to human models, and particularly some of the obstacles or like off-target effects that?
Yeah. Well, to the biggest. So let's see. So there's a few different areas. In the cancer area, it seems pretty clear how to move forward, and we are doing that. And I don't think there are any profound limitations there. Mostly one of the big benefits that the bioelectric field has is that there are already a huge number of human approved drugs that can be used for this. In other words, something like 20% of all drugs are ion channel drugs, and many of them are approved in human patients for other applications. So we in my group, we're working very hard to make sure that what we're using are either or are very close to already human approved drugs for doing this. So that seems that path seems pretty straightforward. In the birth defect arena, which I really actually have a lot of personal interest in. I would really like to solve some of these birth defects. There's a fundamental problem, which is safety testing. It is for obvious reasons impossible to test anything on pregnant women, and so there seems to be this real gap between all the money that goes into developmental biology and occasionally when we actually find solutions to some of these things, then there's this like incredible valley that's very hard to breach, because even if it's very effective in model systems, it's completely unclear how you ever get into human applications, because nobody wants to expose fetuses to these things. So I don't know actually how to move forward on the birth defect side, unfortunately. On the regeneration side, again, I think it's a relatively straightforward path. We're going to do small animal mammalian studies when at Morphoceuticals, which is the company that's licensing this technology, and then presumably they'll do large animal studies and eventually into patients. So yeah, I think it's completely doable. It's just some of this technology is quite novel. This is going to be interesting because these Anthrobots, they're your own cells, and they haven't been genetically modified. Nothing new has been added to them, and they're going right back into your body. So I think this will be somewhat new for the FDA and for things like that. So we'll see how that works out.
Sorry. Okay, one more question over here.
Thanks for your presentation. This is Jason. We interacted, and I'm a huge fan of your work. I actually have two questions, or two things. One, can you really describe like the scope of your work? Because you're talking about this, but I don't think that people know like how many projects and how many teams and how to kind of get involved in amplifying some of this more frontier and pioneering therapies and experiments that you're doing. And my second question is sort of the value proposition for a place like this, where you can have like a right to try environment, and you have a potential like secondary rail to like FDA to speed up these trials and these experiments. Would there be any like possible synergy with developing like say facilities or sending teams over here to do some of this more pioneering work?
Yeah, thank you for that. Well, let's see. The second question, let me do that first. No doubt, I'm sure there are opportunities for collaboration. Now, I'm not a clinician, and so if anybody's interested in moving some of these things forward, we will engage some of the people who are actually working with me to try and move this forward. So, for example, for the Anthrobots and for the aging work, I didn't even talk about any of our aging projects, but for the aging work and for the Anthrobots and for the cell training, there's a company called Astonishing Labs which is licensing this stuff, which would be very interested. For the limb regeneration work, it's a company called Morphoceuticals. For the environmental biorobotics, it would be a company called Fauna Systems, and we would be talking to them. I don't know anything about building facilities out of the U.S. or anything like that, but I'm happy to discuss if there are people who want to try to move this forward. As far as the scope of the work, I mean, we are, let's see, we are, my academic lab is about 40 people roughly. My Allen Center has a number of other PIs besides myself. But my primary appointment is at Tufts. I also have an appointment at the Wyss Institute at Harvard, and we have some work going on over there. But fundamentally, we work across lots of different areas. It may seem very sort of biomedical focused, but actually the thing that holds together all of the different projects that we do is a much more fundamental question about the nature of embodied minds. And so I'm interested in diverse intelligence and ways that mind can exist in the physical universe, which means we do some conceptual work. So I put out a bunch of philosophy papers. We also do lots of computational work, and we also, of course, do the wet lab. And all of these things, the reason that we have these biomedical implications is that we try very hard to make sure that our advances on these philosophical questions are very practical. So when you have better perspectives on the deep questions of life, such as what is intelligence, what is collective intelligence, why are we more than the sum of a pile of a bunch of neurons? What is it that allows systems to align their parts towards higher level goals that none of the parts can comprehend? Once you start having better answers to those questions, it directly leads to technologies. And in particular, we use groups of cells as a collective intelligence, and we try to understand where do the goals and the preferences and the problem-solving competencies of these unconventional collective intelligences come from. And so that leads to, we then have applications in robotics, in AI. We have a number of things coming forward in bio-inspired AIs. We have projects on virtual worlds for living materials, so basically hybrids where living tissues live in a kind of almost like a simulated video game world where you can train them to stay away from epileptic states and things like this. So yeah, lots of different projects, probably on the order of 20 to 30 different projects, all aimed to understand how we can better relate to other unconventional kinds of minds that exist inside your cells, inside your organs, and probably in the ecosystem around us as well.
Okay, we have one more question.
Hi, Michael.
Hi.
Fascinating presentation. So how, I guess I also have two questions. So how would these Anthrobots be delivered, and what would their lifespan be? Would you put in like a timer, or like a metabolic, like kill switch sort of thing? Yeah, how long, I guess, would they last, or like be proliferating in your body post administration?
Yeah, good question. First of all, they don't proliferate, so they do not actually proliferate. As many as you put in, that's how many you have. We don't need to put in any kind of a kill switch or any kind of metabolic limitation because they normally live just a few weeks. So, and my guess is in the body they will live even less long than that. I mean, this right here, what you're seeing here, took about 4 days. And again, this was basically our first experiment, so we haven't even optimized anything about the creation of these bots yet. But fundamentally, yeah, they live a few weeks, and they biodegrade on their own. And we've made, which again I didn't bring here, but we have nice videos of them spontaneously degrading into harmless components in your body. Of your own, these basically just pieces of your own epithelial cells that are shed in your airway by the thousands every day. So the safety profile from that perspective is really good. These things are self-limiting in a very strong way. How do we deliver them? Yeah, we don't have any in vivo data yet, but you can imagine let's say people who currently get shots into their eye for various kinds of retinal diseases. You can imagine very easily introducing these things that way into the eye for, let's say, repair of retina or optic nerve or things like that. You could imagine introducing them into spaces in the spinal cord. You can basically anywhere that you have room. I mean, these things are not, they're multiple cells big. In other words, they're not tiny subcellular things like viruses. They're reasonably large, and so anywhere in the body that there's room for them to move around, in the intestine, in the knee joints, you could imagine them injecting them inside of arthritic joints and having them sculpt the inside of the joint and maybe deliver some pro-regenerative molecules and things like that. Anywhere there's space, you can inject them.
And then also, I guess just a follow up, would there be a benefit to engineering humans to produce like anthrobots just like naturally?
Interesting. Well, I look in the future. I think it's entirely possible to make those kinds of changes, but in the short term, you're much better off. I mean, engineering humans is a kind of a big undertaking, right? And so I'm sure that humans will become increasingly engineered. I have no doubt of that. But we don't have a need for that now. You can very easily either donate a tracheal sample, or eventually we'll just learn to make this out of skin cells or something. And then we can introduce them into your body in whatever amounts you want. So I see your point, and probably the humans of the future will generate all kinds of things like this on their own. But right now, I don't think we need to get into the issue of actually modifying people in a permanent way. I think a benefit of this technology is that it is such a light footprint on. I mean, just much less invasive than any kind of gene therapy or anything like that.
Okay. Any other questions? Oh, one here.
Yeah, you mentioned some thoughts around your work about consciousness and maybe defining different levels of intelligence or different types of intelligence. And I know that is some of your other talks, but could you maybe give people a little bit of an overview of some of the discoveries you've kind of made recently around this space, and maybe introduce that conversation?
Sure. Yeah, let's see. So let's put up just for a second. Let's put consciousness aside for a second and talk about intelligence. The thing is that consciousness raises some very specific and difficult philosophical issues, and there's a lot of work that we can do in unconventional intelligence long before we ever get bogged down into the issues of consciousness. So let's talk about intelligence. One of the things that we've been working on is this idea that we as humans have inherited a lot of mind blindness, meaning that in our evolutionary history, all we needed to do was recognize intelligence of medium-sized objects like us moving at medium speeds in three-dimensional world. So we're pretty good at noticing crows and other primates and things like that doing intelligent things. Sometimes we can even notice whales and things like that doing intelligent things. But that's pretty much it. We're just our intuitions are really pretty terrible as far as identifying unconventional minds. We're not good at noticing intelligent behavior at different scales, in different embodiments, and in particular in different spaces. So something that's very interesting is that while we are all kind of hyper focused on movement in three-dimensional space, and you will see people talk about embodiment, and they'll say, "Oh, this AI is not embodied because it sits there in a server box and it doesn't move around the world." They're viewing embodiment as necessarily in three-dimensional space. They will say the same thing about organoids. Oh, well, this brain organoid in a dish, we don't have to worry about it because it doesn't have a body. It's not a real mind because it's not embodied. But biology actually uses that kind of intelligent navigation of problem space, meaning the perception action loop, This is what agents do. They take measurements of themselves and their outside world. They make decisions based on what they've measured. They have models that they build up over time about what they are and what the outside world is, and they use those models to guide their behavior. Those kinds of things that people study in neuroscience as far as moving around the 3D world are actually ubiquitous in biology. They're everywhere, and it's really much more fundamental. It has nothing to do with moving around a 3D world. So individual cells are already doing all of those things in other spaces. For example, cells navigate physiological state space, so the space of all possible physiological states. They navigate transcriptional space, which is the space of all possible gene expression states, which is a very high dimension, like 20,000 dimensional space. They navigate, of course, cells, of course, navigate anatomical space. So I do a whole talk about how morphogenesis is basically a navigation task of this collective intelligence trying to find its way and figure out where it's going, that where it starts out as a single cell and it needs to reach a particular target morphology. And so what becomes very interesting once we generalize intelligence to the ability to reach your goals in some arbitrary space using some degree of ingenuity when life doesn't go your way, meaning there are barriers, things change, there's novel stressors and so on. It turns out that the same kind of intelligence that we see in the brain and that we're very used to is actually going on all over your body. And that the best way to think about these kinds of things in medicine is not as a bunch of molecular clockwork that you're going to try to micromanage, but actually as different kinds of intelligence with which you can communicate. You can try to reset their goals. You can train them to do things. You can change their view of the environment, their past, their priors, and so on. And so the implications of this are, first of all, the very practical implications is that now all of the tools, not just the tools of hardware rewiring, but the tools of cybernetics, control theory, behavioral science, computational neuroscience, and so the computational psychiatry, actually, all those tools become accessible to help you exert effects in medicine and bioengineering and so on. But the deeper kind of philosophical implication of this is that, for example, I'll just give you one example. For the exact same reason that we think we have consciousness that is associated with our brain, you should take very seriously the fact that all of your other organs meet the same criterion. So, for example, let's ask ourselves how you look at another human being, and you're pretty sure that they have consciousness the way you do. And when I say consciousness, I mean some kind of inner perspective, some kind of valence, meaning they care about what's going to happen next. They have an inner world, and they have some kind of moral importance to them because they can suffer and they can strive for goals and so on. So you look at another person, or you might be looking at an alien visitor from another planet, and you want to know whether you believe that they have a mind like you, of maybe not like yours, but another conscious mind. So there's a few pieces of evidence that people usually take. First is behavior. Do they behave as though they had a mind? Meaning, do they exhibit goal-seeking behavior? Do they appear to like and dislike certain things? Are they intelligent in that they can solve problems and so on? If those are your criteria, those are amply met by all kinds of tissues and organs in your body, just not in three-dimensional space. They do it in other spaces, and that seems perfectly reasonable. And again, when you meet an alien intelligence, there's absolutely no guarantee that their life takes place in the same kind of space that yours does. Then some people say, well, okay, behavior isn't really good enough because maybe it's just faking. Maybe it's doing behavior, but there's, as people say, there's no one in there. It's just going through the motions like an automaton of some sort. Well, okay, so if it's not behavior, what is it then? Well, then some people think it's evolutionary conservation, meaning that this creature uses the same mechanisms that I use to maintain my consciousness. Well, if that's your criterion, then again, all of the organs in your body fit because they are using exactly the same mechanisms that your brain uses. In other words, ion channels, electrical synapses, neurotransmitter machinery. All of that stuff is way older than brains, and it exists in every cell in your body. So that's not it. That's not a distinguishing difference either. And there's a few other things we could go through, but basically, at this point, there is no obvious convincing reason to say that the kind of thing that goes on in your brain is also not going on in the other parts of your body. Now, at this point, some people usually say, well, wait a minute, I don't feel like my liver is conscious. well, right, but neither do you feel that I'm conscious, nor do you feel any consciousness about your non-language using hemisphere in your brain. In other words, it's all well and good that one of the hemispheres in the brain has the linguistic ability to proclaim its own consciousness, but other than language, which true enough, we've actually not found yet anything that we could reliably claim was a proper generative language with a grammar and all that in outside the brain. So other than its ability to convincingly tell stories about its conscious experience, there is no reason, to my knowledge, to think that the other parts of your body are not having some kind of conscious experience as well. So that's one kind of a place where these things intersect with the problem of consciousness. And then there are many other things I could talk about, all kinds of other things that are not in your body that also appear to have the kinds of things we associate with cognitive processes. But that would, that would be a separate talk, I think.
Okay, so we might have time for one last quick question if anyone has a quick question. Okay, we have one last question.
Thank you. I really enjoyed the experiment with the frogs where you're rearranging the facial features and then it autonomously arranges it back. It got me wondering, I don't know much about IVF, but I understand there's the biopsy of the embryo that is required to do genome sequencing. I wonder if you have any thoughts on particular risks of having that removal of matter in the embryo for its development over time.
Yeah, very interesting question. So I don't know about risks in the sense that I don't know the epidemiology of it. I assume people have studied whether that kind of procedure ends up with, I don't know, some kind of subtle, I'm making this up, but maybe some immune issues later or something. I don't know about the specific risk of it. What I do study is the flip side of it, which is the mechanisms that enable a system like that to work in the first place. I mean, isn't it remarkable that you can take an early embryo, literally cut it into pieces, and each piece, instead of giving you a half embryo, what you get is perfectly normal monozygotic twins and triplets and so on. In fact, that's not just through IVF, but that's naturally where multiples come from, is that the early embryo splits in half and you don't get half bodies the way you would with any of our current machine technology, where if you cut it in half, well, that's that. In these cases, each of the systems has exactly this kind of plasticity. And I agree with you, it's exactly like this, where it realizes that half the body is missing, reconstructs it and remodels everything and so on. In fact, people talk about, I think it's funny that people talk about the planarians and how amazing they are at regenerating when at least half of the human population is able to regenerate the entire body from one cell. In other words, I think development is basically just regeneration. That all of this is the ability to correct your shape from different starting positions, and being down to a single cell, the egg, is just an extreme form of regeneration. There's something else to say about this, which goes back to this previous consciousness and also actually back to the genetics question, which is interesting. Just imagine you have a group of cells. Let's say an early embryo is like this flat, at least an early amniote embryo is like a flat blastodisc of, let's say, 100,000 cells, and we look at it and we say, oh, there's an embryo. Well, when you say it's one embryo, what are you actually counting? I mean, there's 100,000 cells. What are you counting as one embryo? And what you're counting actually is you're counting in essence, you're counting a worldview, a story, a commitment of all of those cells to the same journey in anatomical space that they're going to take together. In other words, they're all going to cooperate to build a specific structure. And the fact that they've all bought into the same view of what they're building, that meaning the pattern memory that guides them is they've all bought into that's what it's alignment. It's alignment towards one goal, and that's what you're counting. Now, what you can do, and I used to do this in duck embryos in grad school, is you can take a little needle and you can make some scratches in that blastoderm. Let's say you make 4 scratches. Well, for the next 4 to 8 hours before those scratches heal back up, each of the little islands, right? So you make a bunch of scratches, you generate some islands that are unconnected with each other. Each of those islands is now unaware of the presence of the others, and it self-organizes into a perfectly good embryo. And then eventually they either heal or don't. And if they do, you get conjoined twins. And if they don't, you get separated, regular separate multiples. But either way, you get multiple full individuals out of it, and then they grow up and they become, individuals. Just like in the human case, you would get twins and triplets and so on. So this is, I think, philosophically interesting. The number of individuals that are present within an embryo is not known. And it could be anywhere from 0 to half a dozen to probably more. If I recall, the record is in the 60s, I think, for how many you can get out of a single embryo. And so this goes back again to the genetics problem, because you would think that the one thing that the genome would specify is how many individuals are present in an embryo, and it doesn't even do that, because what it actually specifies is a much more clever thing, which is this dynamic. The vocabulary fails us. It's a dynamic pool of potentiality from which multiple beings, including humans, can come from, and can be anywhere from 0 to however many. And all of them are created on the fly by this process of problem solving that aligns the parts, meaning the cells, towards a larger scale goal using the tools available. Meaning, however many cells you have, you're going to be an embryo more or less. And so, yeah, so I think IVF and in general, I mean, IVF is what they do now for humans and prize cattle and so on. But the basic phenomenon in developmental biology that embryos are actually self-repairing systems, and that you can do these kinds of things, goes back well over 100 years. I think it teaches us something quite important about what we are. We're this dynamical construction that really relies on our parts buying into a specific story, and this goes quite a bit further into biomedicine, where you can, and in fact we are addressing, for example, failure to regenerate as the inadequacy of such a story that the cells at the wound have adopted, and the idea would be to shift them to a different story in which the path towards regenerating becomes actually seems accessible to them. And the same thing for cancer. These cells have basically rejected the collective, the memories of the collective in terms of building organs, and have struck out on their own in a much smaller cognitive light cone. So all of this is again like this kind of interesting place where deep philosophical questions really impact on very specific aspects of bioengineering and medicine.
Okay, Michael. So thank you so much for coming with us here today. It was an honor to host you here in Infinita City, and I hope that next time we can have you here in person.
Thank you very much. I appreciate that. Yeah, I appreciate that a lot, and thank you for having me. Some great questions. Thank you for the discussion.
Perfect. Thank you so much, everyone. Round of applause.
Thank you all. Have fun.